I think in myeloid CAR development, the challenge is there's not going to be a single antigen that is the optimal target. So we don't have CD19 in myeloid malignancies. And so the way CYAD-01 gets around this is that you're putting NKG2D, which is the predominant killing receptor of NK cells, in a T cell. And it's really agnostic to disease state. So many cancers, including AML and MDS, upregulate ligands for NKG2D, MICA and MICB being sort of the main ones, although there's the ULBP family. And so that, I think, is the overall goal, is that so we don't have an optimal antigen, is it better to be a little bit more non-specific? And so we have had some very exciting results as far as a couple of patients that have had a remission from the therapy, have been bridged to transplant. And one of my personal patients is now like 3 years out from the original therapy. I think the challenge has been, although we do see activity with blast reductions and some patients responding, is that even despite some of the change in the manufacturing, even with the addition of lipid-depleting chemotherapy, we've not been able to improve the durability of responses. And so yes, there is activity, but I would say not enough at this time. And so I think actually what's more critical is there's a second generation product, or CYAD-02, which was a poster at ASH, where part of the issue may be these CAR T cells may actually kill each other. So inflamed or activated T cell can upregulate the ligand MICA, MICB. And so to get around that, the second generation product uses short hairpin RNA to knock out or knock down MICA, MICB in the product, which may improve expansion and persistence. And at least in very early data, the clinical efficacy look quite interesting. So I'd be looking more forward in the CYAD-02 product than with CYAD-01.